To make aseptic transfers, work close to a Bunsen burner with a hot flame so that the updraft of heated air prevents microorganisms from settling on your plates. Never leave cultures uncovered after you've examined or used them. Remember to adjust the Bunsen back to a luminous flame when you've finished using it to avoid the danger of setting fire to yourself. To transfer bacteria from an agar plate to a liquid culture, first sterilize a wire loop by heating it in a Bunsen flame. Hold the loop upright in the flame so that the whole length of the wire gets hot. Cool the loop by touching the side of the agar plate. If you don't do this, any bacteria you touch will be killed by the heat. Pick up a small amount of material from the surface of the plate. To inoculate a liquid culture, work close to the flame. Hold the lid of the bottle or flask with your little finger, don't put it down on the bench, and pass the mouth of the bottle through the flame. Wash the material from the loop and then immediately pass the mouth of the bottle through the flame again and replace the lid. Flame the loop again to sterilize it before you put it down or use it for another transfer. To pick up cells from a liquid culture, use a sterile flamed loop to pick up a drop of liquid. This is usually sufficient to inoculate an agar plate or to transfer to a microscope slide, but sometimes you may need to repeat this procedure 2 or 3 times to make a smear for microscopic examination. To transfer larger volumes of liquid, use the Gilson pipettes. Attach a sterile tip to the end of the pipette and push the plunger down to the first stop position. Put the tip into the liquid and slowly release the plunger. Put the filled tip into the vessel where the liquid is to be transferred and press the plunger down all the way to expel the liquid. Eject the tip into the appropriate waste container. The ability to streak out bacteria on agar plates in such a way that you obtain single, well-separated colonies after the plate has been incubated is a fundamental skill in microbiology. Unless you can do this, you won't be able to isolate and purify bacteria from mixtures. As demonstrated here with a felt tip pen, the principle is to draw out a small amount of material across the surface of the agar. At each stage, ever-decreasing amounts remain, so that by the end of the process, very small numbers of bacteria are left. To begin, sterilize a wire loop, then cool it and pick up a small quantity of material from a plate or a drop of a liquid culture. Place this on one side of a clean agar plate and with a circular motion make a smear of the material. Flame the loop and cool it on the agar so that you don't carry over too much material from the inoculum. Then make 3 or 4 streaks along one side of the plate, drawing out material from the smear. Flame and cool the loop again, and at an angle to the first set of streaks, make another 3 or 4 streaks along the side of the plate. Keep the loop in contact with the agar, but lift it off between each streak. Don't use too much pressure or the loop will dig into the agar and wipe off the material it's carrying. Repeat the same procedure for a 3rd time. Finally, flame and cool the loop and make one long continuous zigzag streak across the plate without touching any of the areas you've already streaked. If the procedure is carried out properly, dragging the loop across the agar will deposit isolated single cells. Incubate the plates upside down to prevent condensation dripping on the surface. After the cells have grown, and if you've streaked the plate correctly, you'll have well-separated colonies which you can identify and use for further experiments.